Hello everybody, welcome to our video. Today we are talking about biochemistry chapter 6, which is DNA and biotechnology. So let's get started. Okay, so first thing in this chapter is just a review of nucleic acids because those are the basis of our DNA. So you have nucleic acids, which are polymers of nucleotides, okay? And they have a primary structure, which is the linear sequence of nucleic acids. Then we have the secondary structure, which is the interaction of a base between the same molecule. We have the tertiary structure, which is its location in 3D space. And then we have the quaternary structure, which is the interaction of nucleic acids with other molecules. So there are 4 levels of nucleic acid structure. And we do have this for amino acids as well. And then, it's the same thought process, but just make sure you look at the details in this and you're able to, differentiate the two. So we have DNA, which is deoxyribonucleic acid. RNA is ribonucleic acid. Okay, so the deoxy part is important. And I forgot to draw it here, but even pause this video right now and in another tab, look up these 2 images. So look up an image of RNA and look up an image of a DNA molecule and see how the DNA is different because the oxygen is gone, the deoxyribonucleic acid. Okay, so just so you have that visual and you can recognize it, that's super important and I'm definitely meant to write it on here. So in DNA, we have a nucleoside, which is a 5-carbon sugar and a nitrogenous base. Okay, and a nucleotide is a nucleoside, so our 5-carbon sugar and a nitrogenous base with phosphates attached. So that's what makes it a nucleotide versus a nucleoside is the phosphate. So nucleotides include adenine, thymine, guanine, and cytosine, and uracil, which replaces thymine in RNA. That's how you can differentiate if you're looking at RNA or DNA. If you have uracil, you know that you have an RNA strand. If you have thymine, you know that that is a DNA strand. Okay, so just a little bit of background on DNA. We have Watson and Crick discovered that the backbone of DNA is made of alternating sugar phosphates, okay? DNA is read from the 3' to 5', and I'm going to show you that on a diagram in a moment, but just keep that in mind, make note of it. It's read from 3' to 5', but it's built from 5' to 3'. And we'll look at that in a moment. But the 2 strands of DNA are anti-parallel, and they're wound in a double helix, okay? And the nitrogenous bases, we have purines and pyrimidines. So the purines are adenine and guanine, and those have two bonds between them. So if you have adenine and guanine, there's 2 bonds connecting them. If you have pyrimidines, which are cytosine, uracil, or thymine, those are kind of a 1-ring structure, and they have 3 bonds, so those are stronger. A bond with pyrimidines are stronger, okay, versus purines that only have 2 bonds. So take more energy and more heat, higher temperature to disrupt the bonds between pyrimidines, so between thymine, uracil, and cytosine, compared to between adenine and guanine. So the other thing about their structure, so purines are 2 rings, and pyrimidines are just a single ring. And so I also wrote something on this side. Let me move over here. So I know it's kind of hard to see at the top, so I'm just going to read it really slowly. So this is hydrogen bonds are used between nucleotides of other strands, okay? Where phosphodiester bonds are used between the sugar and the phosphate of the same strand. So again, this goes along with looking at a picture of DNA. Pull it up right here, pause the video, in another tab, look up a DNA strand at the molecular level, and you'll see hydrogen bonds between nucleosides of another strand, nucleotides, sorry, of the nucleotides on another strand, and phosphodiester bonds between the sugar and the phosphate when you're looking at the same strand. That's super important. Okay, so another just couple quick facts that are important to know. So we have 46 chromosomes in humans. DNA is in eukaryotic cells is wrapped around histones to form nucleosomes, which is then stabilized by other proteins. So you don't just have all the DNA flying everywhere. You have the DNA that's wrapped around histones, and a group of histones is called a nucleosome. And that just helps us compact our DNA. So you have chromatin. You can have heterochromatin or euchromatin. So heterochromatin is when it is densely packed, and when the DNA is densely packed, that is silencing those genes. But if you have a euchromatin, that means that it's uncondensed, and those genes can be expressed because you can get our other things that we're going to discuss. You can get those things to replicate the DNA and to produce mRNA, which we'll get into. But you can get those factors. I'm trying to think of the word. Translation, transcription factors, all those, you can get closer to the DNA when it's uncondensed, which is why when it's uncondensed, it can express the DNA and the genes. Whereas if it's tight and silent, our transcription factors cannot get to the DNA. So you will not see those genes expressed if they're condensed so tightly. So we have recombinant DNA, which is when you have nucleotides from two different sources. We have hybridization, which is just the joining of complementary base pair sequences. We have, when we're looking at experiments, you have transgenic mice and knockout mice. So transgenic mice introduce a gene of interest into the genome, whereas the knockout mice are created by deleting a gene. Knockout, deleting the gene, you can kind of remember that. But those are some of the vocabulary you might see in different passages when they're conducting an experiment on, gene mutation, regulation, things like that. So we also have DNA repair. And DNA repair, I'm sorry, I'm just saying now that it's super light, some of these markers. I need some new markers, y'all. But I'll read it out for you. So DNA repair, there's a lot of stuff underneath that. So we have oncogenes, which are different from mutations, and oncogenes promote cell cycling, and they may lead to cancer. Tumor suppression genes code for proteins that reduce the cell cycling, okay? Because when we know from previous chapter, cancer cells like to replicate, right, uncontrollably. So when you have a tumor suppression gene, that's trying to reduce the cell cycle of that cell. We also have proofreading, which is done by DNA polymerases, and they take out the incorrect match pairs. So we'll look at that when we look at their DNA replication, but that is definitely an important tool when during DNA replication is proofreading by the DNA polymerase. Then we have mismatch pairs, which occur in G2 phase, using MSH2 and MSH1 genes. Now this, again, we'll look at in a later chapter, but just keep in the back of your mind that you have mismatch repairs, okay? Then we have base excision repairs, which fixes non-deforming lesions, such as cytosine deaminations, by removing the base. So that's just, a very specific example of a base excision repair. So excision, taking away. So you're removing the base to repair. Then we have nucleic excision repair, which fixes a helix deforming lesion of DNA, and so it cuts the DNA and then patches the process and patches. So that is nucleic excision repair. So the words kind of tell you what the process does, but you should just remember that that's under DNA repair. So now for the last part of this video, I have another